Hi, I'm Peter Cook. Welcome to the pan-genomic model, the role of transcription factories in chromosome structure and function. I have given many talks on the model, and here I give some answers to frequently asked questions that provide more detail than in the main presentation. First, a word of caution. Because of the prefix pan in pan-genomic, and because I give an alternative view, questioners often expect me to explain everything. Of course, I cannot. So in my answers, I try to focus on the unique insights offered by the model, but nevertheless try to point out what we still don't know. There is an audio overview at the beginning of each FAQ without a video of me. So please could you control slide changes, and then you can read at your own pace, as answers are given mainly as text. All these structures fit perfectly into our model. Recall that in this context, CTCF may be fashionable, but it is not a big elephant in this room, as it is not encoded by plants or bacteria that nevertheless have TADs and associated boundaries. And so I only discuss CTCF in passing. At the end, I discuss high-resolution methods that are detecting exactly the contacts expected of factories. They fit perfectly into our model. And I will counter with another question. What is the difference between a transcriptional condensate and a factory? The short answer is they do cluster. The long answer is interpretation depends on the technical detail, and I'll illustrate some of the complexities. I begin by considering polymerases productively transcribing typical genes. In this case, many people believe that an active bacterial or mammalian gene is transcribed by many polymerases at any particular moment. But the evidence is against this. Then I consider non-productive transcription, which seems to occur much more often. There is lots of evidence. I begin with some that is old and circumstantial. That doesn't mean it is less compelling, and progress to some that is more recent. I also discuss a related question. If active polymerases are immobile, are factories static structures? We have never said they are, because they clearly come and go. I also end with obvious caveats about space and time. At least 92% of transcription by mammalian RNA polymerase II occurs in factories, so probably all. The rider is that most cars are made in factories, but then I have seen one or two homemade ones. I also add a counter question. How much transcription goes on in the transcriptional condensates and hubs that are so fashionable? They cannot be transcribed simultaneously according to any model, and ours provides a simple explanation of a mysterious phenomenon known as transcriptional interference. I take energy inputs in a broad sense to include those involving both ATP hydrolysis and binding energies, and so on. Unfortunately, we know little about ATP requirements for most cellular functions, so I can't add much insight into those involved in building factories. However, simulations show that you don't need additional ATP inputs to cluster the transcription machinery, but you do need it to ensure components in clusters exchange rapidly with a soluble pool. I guess that evolution stuck with that. It was thought that splicing occurs post-transcriptionally. Now, most is known to occur co-transcriptionally, and so in factories. And there is good circumstantial evidence for this. Unfortunately, I don't have a precise answer, but I can provide a framework to think about this problem. The short answer is surprisingly little, but I have to add that the little we do know is still much more than what is known about transcription hubs and condensates. We only have old estimates. These should certainly be revisited. Yes, but because trans contacts are detected poorly by high C, and if detected are generally discarded, they have been underestimated. Structural and functional changes occur so quickly during mitosis, it is difficult to establish cause and effect. Moreover, results are often conflicting. For example, loops detected using nuclease digestion remain, whereas those detected by high C are lost. So lots of what goes on remains mysterious. I wish I could supply an accurate answer. A lot depends on how you define loops. We don't know, but here are some ideas. I don't know, but here are some speculations. Mechanisms underlying both these processes are mysterious, with a bewildering array involved in meiotic chromosome pairing in different species, so uncovering universal principles is challenging. But I think the explanations I offer here are the most plausible available. I both agree and disagree. I do try to justify why it's worth trying for a model as simple as the pan-genomic one. Thank you for your interest, and I do hope you enjoyed the journey.